Okay, welcome back to our notes on cell membranes and how particles move across. First thing we need to talk about is basically most movement across a membrane follows a concentration gradient. So if you remember from our xylem and phloem, that concentration gradient is how molecules moved through that type of vascular tissue. So molecules go where they are needed. They go where it's a low concentration. Before we can get into those details, we need to learn how to calculate percent concentration. The definition of concentration is how much stuff is dissolved. And the formula for that is the amount of stuff, or the solute is the stuff of what goes in, divided by the total volume of the substance times 100 equals a percent concentration. So for example, a 10% concentrated solution has 90 grams of stuff or liquid, 10 grams of solute. When you combine those, it's a total of 100 grams of substance, but you had 10 grams of stuff in that total. So percent is always part over the whole times 100. The part is whatever's less in amount, so the 10. The total is everything altogether. Multiply it by 100 to turn it into a percent. So here's another example. I have, that's the wrong one. I have 50 milliliters of water right here. I'm going to add 50 milliliters of stuff, and that gives me a total volume of 100 milliliters. It's 50% concentrated. Okay. Here's another amount. There's 90 milliliters here, and I add 10 milliliters of stuff. This is a 10% concentrated solution, okay? So, hardest thing for people to remember is this bottom number is total volume. You have to add the liquid and the amount that's going into it, and then the number on top is what you added in. So, now that we know how to calculate concentration, we need to define them. So there are three words you need to know: hypertonic, hypotonic, and isotonic. If something is hyper, the prefix hyper means high, so it's a high concentration solution, which means it has more solute than what you're comparing it to. Hypo is low, so hypo means below. A hypodermic needle goes below the skin, and then iso means equal. So this is what this means. Here's where people get confused. This is a really strong concentration, but I can't call this hypertonic unless I'm comparing it to something else. So this by itself is 50% concentrated. This by itself is 10. If you look at this, you might say, well, that's a high concentration, but you cannot use the word hypertonic unless you're comparing it. So by itself, this is 50% concentrated, but when I compare it to this one, this is hyper, this is hypo. So you can't use the word hyper unless you have something next to it that can be hypo, and vice versa. I can't say this is a low concentration. The only thing I can say about this by itself is that it's 10% concentrated. But when I compare it to this, I can say hypo and hyper. Again, the word iso can only be used if it's equal. So if I take this beaker, this is 50% concentrated, and I put it over here, it doesn't matter that there's more here or less here. These are both 50% concentrated solutions. They are isotonic to each other. So these three words on this slide, you cannot use alone. They have to go with something else, okay? And even though there's less stuff over here and more here, because the concentration is equal, they are both iso. So you might look at this and say, well, that's a lower volume. It has to be hypo. It has nothing to do with total volume. We're looking at concentration. So there's 50% concentrated here, 50 here. They are equally concentrated. All right, so we're going to keep doing that same thing to get it in your head. So there's three pictures here of cells. This orange area is inside the cell. This is outside the cell. So you need to look at how much stuff is on each side. And we're going to assume that water is the same. So how many particles are here versus here? If I labeled this cell, I would say outside is hypo, it's low concentration. Inside there's more stuff, so it's high concentration. Right here I have 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So those have equal concentrations on both sides. I have to say both sides are isotonic. And this third slide, there's clearly more stuff outside. So outside is hyper, inside is hypo. So that's how we use those words. Okay, so how do particles move? Based on that, there are 3 ways particles move across a cell membrane. Well, actually 2. We'll break it into 3. The first way is called diffusion. When something diffuses, it follows the gradient, meaning it goes where it is needed and energy is not used. That is the same concept of taking this beaker of water. I'm going to drop in some food coloring. That food coloring moves where it's needed. Now, it might take a while because of the temperature of the water, but eventually my whole entire beaker will be the same color because those little green coloring particles go where they are needed, and they move where they are needed until everything is equal. So I did not have to add energy to this. That is diffusion, where particles are going where they are needed. So I'm going to set it down, and I'll prove it to you in a minute after it takes some time. There are 2 types of diffusion. So again, remember diffusion follows the gradient, doesn't need energy, simply goes where it's needed. It's getting better up here. You can see it's going where it's needed. So if substances are super small and they can slip right through the membrane, that is called simple diffusion. Sometimes molecules are bigger, or sometimes they are repelled by that membrane. That's called facilitated diffusion because they have to go through a door. And then the second main way, or third, is active transport. Active implies energy. When something is moved through active transport, it actually goes against the gradient. So you would push particles in where they are more crowded. All right, so let's look at these different types. So again, simple diffusion. Molecules go where they are needed. I encourage you to open up this presentation and watch the animations, but they're going to show you. So here's a lot of particles, here is none. Nature wants to be fair. Those particles can slip right through the membrane or right back out. So right here we have a hypertonic environment going into a hypo, they'll move down. Here you have, so it's showing you right here, it's hypertonic moving to hypo. As soon as things are balanced, then they're isotonic and the particles actually move both ways. So motion never stops. Particles are moving in and out of your cells all the time. First they move where they're needed. Once it's balanced, they'll go equally back and forth. This only works for small items or if they are non-polar. They have to match this lipid membrane. Remember, these fatty tails don't like water. Water is charged, water is polar. So polar items or anything with a charge will not get through the membrane. But things like food, oxygen, carbon dioxide do not have a charge on them, so this oily membrane lets them slide right through. So in order for simple diffusion to take place, you have 2 requirements. You have to be tiny, like a gas, and you cannot have a charge. Okay, if you are charged or fat, you don't get to go through the membrane because number 1, you can't fit, and number 2, this non-polar lipid membrane will kick you back out. Again, once equilibrium is reached, life never stops moving, so the particles keep moving back and forth. All right, so let's look at that same picture. We know how to label these environments, so which way will particles move here? Are particles going to go out of the cell or in? And if they follow the gradient, they go out into the lower concentration. Here it's equal, they're going to go both ways. And here particles are going to move into the cell or into where they're needed. Again, if these are super tiny, they get to slide right through the membrane, no energy is used. Okay, there's another type of diffusion. It's still called diffusion because it doesn't need energy, but this is how larger items or charged particles move through. So if you are charged, this polar membrane, these lipid tails are going to kick you out. If you're too big, you're not going to fit through the little slots in between these little phospholipids. So this is when we have to use a protein door. Those protein channels that are embedded in the cell membrane will let those molecules through, but it works the same way. They're still going to just slide right through and go into where they're needed. The only difference is they have to go through a door, or they need to be helped or facilitated through. All right, so let's look at the same thing here. Same pictures. I put this little door in. The same thing happens, all right? We can still label the cells the same way, but this time particles go out through a door. When it's isotonic, they go both ways, but just through that door. And if they're moving into the cell, they still come in, but again through a door. So diffusion is always low energy or no energy. It always goes where particles are needed. If it's simple, it's because they're tiny and they match the membrane or they're nonpolar. If it's facilitated, it's because they're bigger or they are charged so they can't go through the membrane. They have to go through a door. Okay, just to make it a little bit weirder, osmosis is diffusion of water. So water's constantly moving in and out of your cells. We're mostly made of water. The difference is water is charged. Water is actually repelled by that lipid bilayer. So even though it's a tiny molecule, it can't slip through. It has to go through a doorway. Also, when we're talking about water, water goes where water is needed. So if you look at this picture here, there's lots of particles right here. There aren't a lot right here. The particles are going to go where particles are needed. Water will go where water is needed. So in this example, this membrane that's in between, these particles can't fit through. They're too big, and there's no doorway for them. So water will even out the concentrations because water will go to the other side. So osmosis is a very specific type of facilitated diffusion, talking about water moving and water going where water is needed. So let's look at this. If this was an osmosis example, there's more stuff here, less stuff here. Okay, water is going to go into the cell. The water goes where it's needed and the particles stay. If it's even, water goes back and forth equally. I should have put the arrows here. And if there's more particles outside the cell, water goes out of the cell. So particles go where particles are low. Water goes where water is low. But here's where it gets weird. The area where water is low is actually hypertonic because it's high concentration of stuff. So I remember it this way. Hyperactive kids, think about this as a hyper environment. There's lots of activity here. If somebody was hyperactive and running through the classroom, we would probably throw water on them to calm them down. So water gets thrown into the hypertonic solution to try to calm it down. Okay, so we'll go through this in class, but particles go where particles are needed, water goes where water is needed, and it's always opposite. The particles are needed outside of the cell, but water is needed inside the cell. It's both types, both of them are types of diffusion. Osmosis has to be facilitated because water is charged. This happens in real life. If this is an animal cell and it's in a hypertonic solution, so there's a lot of salt, water will leave the animal cell or the plant cell. This is when we saw those onion cells shrink up because they are in salty water. If your cells are in a even solution, it goes back and forth. If your cells are in a low salt solution, water will go into the cells where it's saltier, and then your cells can actually explode. So this first example would be a picture of you just drank a lot of salt water, okay? You were thirsty, but you drank salty water. All the water leaves your cells, and you actually get dehydrated. That's why you die if you drink ocean water. Here is a solution where you actually drank too much regular water. So if you OD on water, your cells would actually explode because you would have so much water out here that it would be hypertonic in your cells and water would go where water is needed and you could actually die from drinking too much water. We'll talk about it later. Okay, the next 3 slides here are the last page of your notes. You need to practice identifying the concentrations and telling me which way water will move or not. So we will do that together in class or you'll have to look at a key, I guess, if you're not here. Sorry, try to come to school or email me with questions.